For years, gene editing felt like something straight out of a movie. But imagine if we could edit the code of life, like uh, fixing a typo in a Word document, cut, paste, fix errors in DNA. Well, that's not science fiction anymore. Today, we're talking about CRISPR-Cas9. It's a technology so revolutionary, it actually snagged a Nobel Prize back in 2020. And it's changing, well, almost everything. Medicine, farming, our basic understanding of life. So this deep dive, it's our journey to really get under the hood of this amazing tool and figure out what it means for all of us.
Yeah, it's genuinely a massive shift. I mean, a real paradigm change in biology and medicine. Before CRISPR, messing with genes was incredibly clunky, really difficult, expensive. Now we have this tool that's precise, it's accessible, it lets us get right in there at the genetic level. And understanding this, it's not just for scientists in labs, it's kind of crucial if you're curious about where health, maybe even humanity, is heading.
Okay, let's unpack this CRISPR-Cas9 then. Starting right at the beginning, its origin story, which is pretty surprising. The name itself sounds a bit intimidating, doesn't it? Clustered, regularly interspaced, short palindromic repeats. Wow. But the idea behind it is actually, well, kind of elegant. So where did this system first show up?
Right, it sounds complex, but the discovery, it wasn't actually scientists looking for a gene editor for humans.
Right.
Not at all. It was found in bacteria and archaea, these really ancient life forms. And for them, CRISPR is basically their immune system, a defense against viruses trying to invade them. Think of it like this. Bacteria sort of take a genetic mugshot of a virus that attacks them. They snip out a tiny piece of the virus's DNA, and they stitch it right into their own genome, into these CRISPR regions. It's like a genetic memory card of past infections.
Huh. So the bacteria keeps a log, like a little library of dangerous viruses it's encountered. So what happens when that same virus shows up again? How does it fight back using that stored info?
Exactly. So when that known enemy reappears, the bacterium is ready. It transcribes those stored viral DNA snippets into RNA, creating what we call a guide RNA. This guide RNA then partners up with a CRISPR-associated protein. The famous one is Cas9. Now, the guide RNA acts like, well, you could say like GPS navigation. It leads the Cas9 protein, which is like a pair of molecular scissors, directly to the matching DNA sequence in the invading virus.
So the guide RNA says, here's the target.
Precisely. And once the Cas9 finds that exact spot on the viral DNA SNP, it cuts the viral DNA, disabling the virus. It's incredibly specific, this defense mechanism. It gives bacteria a kind of adaptive immunity.
That GPS and scissors analogy really helps visualize it for bacteria.
Yeah.
But okay, how did we take that ancient defense system and turn it into something we can use? A tool to edit genes in, say, human cells.
Yeah.
What are the key pieces we need?
Right, that's where the human ingenuity comes in, adapting this natural system. It boils down to four main components that make it work for us. First, you've got the Cas9 protein itself, those molecular scissors we mentioned. This is the enzyme that actually makes the cut in the DNA double helix. That precise cut is what starts the whole editing process.
Okay, so Cas9 does the cutting, but how do we tell it exactly where to cut in, like the huge human genome? That's where the guide comes in, I guess.
Exactly right. Component number 2, the guide RNA, or gRNA. This is the programmable part, the real genius bit for us. We designed this RNA molecule in the lab. Its sequence is made to perfectly match the specific DNA target we want to edit. So it literally guides Cas9 like that GPS, again, straight to the right address in the genome. And the beauty is we can design a guide for almost any gene sequence. It's incredibly versatile.
So the gRNA points Cas9 to the right spot, cut here, but genomes are huge. How does Cas9 make sure it only cuts there? What stops it from, accidentally cutting somewhere similar but wrong?
Oh, an excellent question. And that leads us to the third absolutely critical component, the PAM sequence. Stands for protospacer adjacent motif. It's just a very short, specific DNA sequence, usually NG2, for the standard Cas9. And it has to be located right next to the target site on the DNA strand. Cas9 first looks for this PAM sequence. If it finds it, then it checks if the adjacent DNA matches the guide RNA. If both the PAM is there and the sequence matches the guide, then it cuts. No PAM, no cut, even if the guide matches perfectly. This was a crucial discovery. It adds a vital layer of specificity, making sure the scissors only cut where intended. It drastically reduces off-target cuts compared to older methods.
Oh, okay. That PAM is like a safety check then, a necessary landmark.
Yeah.
So Cas9, guided by the gRNA, finds a target next to a PAM, and snippet makes the cut. What happens to that broken DNA? Does the cell fix it?
It absolutely tries to fix it. And that's the fourth key element, the cell's own natural DNA repair mechanisms. We
Okay.
actually hijack these repair pathways. There are two main ones we exploit. First is non-homologous end joining, or NHEJ. Now, NHEJ is the cell's sort of quick and dirty repair crew. It basically just sticks the two broken ends back together, but it's often error-prone. It might accidentally insert or delete a few DNA letters, base pairs at the cut site while patching it up. We can actually use the sloppiness. If we want to disable or knock out a gene, we make a cut, let NHEJ fix it badly, and that often introduces a mutation that breaks the gene.
Okay, so NHEJ is good for breaking things. What about the other pathway? Is that how we can actually correct a gene or insert something new?
Exactly. That's homology-directed repair, or HDR. This one is much more precise. It uses a template to guide the repair. So if we make the cut with CRISPR and we simultaneously provide the cell with a piece of template DNA, a template that has the sequence we want to see there, maybe the corrected version of a mutated gene, or even a whole new gene segment, the cell can use this template during repair.
Mm.
It essentially copies the information from our template as it fixes the break. This allows us to precisely rewrite the genetic code at that specific spot, paste in a correction, or insert a new sequence. That's the really powerful editing part.
So it's this incredible combination then. The Cas9 scissors, the custom gRNA directions, the PAM safety check, and then using the cell's own repair team, either NADA for breaking or HDR for fixing and pasting. That really clarifies how it works. And this combination, this is what made CRISPR-Cas9 such a massive leap forward, right? How did it change the game compared to what scientists had before?
Oh, absolutely. It's hard to overstate the difference. You see, gene editing itself wasn't brand new when CRISPR came along. Scientists were already using tools, things called zinc finger nucleases, ZFNs, and TALENs, and they worked. They could edit genes, but they were just so much harder to use, incredibly slow to develop, astronomically expensive, and really complex to engineer for each new target gene. It often took labs months, maybe even years, and a lot of specialist expertise just to make one working tool.
So CRISPR wasn't just an improvement, it sounds like. It completely changed who could do gene editing and how fast. It like democratized it.
That's the perfect word, democratized. The sources consistently highlight CRISPR as being cheap, fast, and precise.
Hmm.
Suddenly, gene editing went from being this highly specialized, resource-intensive technique to something that pretty much any molecular biology lab anywhere in the world could start using relatively quickly and affordably. And crucially, it works across the board in any organism, plants, animals, humans. This versatility just blew the doors open for research. In my own field, experiments that used to be incredibly laborious, taking months, can now sometimes be set up in, well, days or weeks. That acceleration is just phenomenal.
And that massive impact, that democratization, it didn't go unnoticed, did it?
Not at all. It led directly to the 2020 Nobel Prize in Chemistry for Emmanuel Charpentier and Jennifer Doudna. Their work was specifically cited for developing CRISPR-Cas9 as a gene editing tool, recognizing they'd get in the world something that fundamentally changed how we do biological science.
Incredible. From bacteria fighting viruses to a Nobel Prize and a tool labs everywhere can use. Okay, let's talk practicalities. What are the actual tangible ways this is being used right now? How is it impacting, us?
The applications are already incredibly diverse and growing constantly. In medicine, it's hugely promising. We're seeing clinical trials, for example, for genetic diseases caused by single gene mutations, like sickle cell anemia and beta thalassemia. The idea is to take a patient's blood stem cells, use CRISPR to correct the faulty gene, and then return the corrected cells to the patient. Early results are looking very encouraging. Then there's cancer therapy. CRISPR is vital for engineering CAR cells. These are a patient's own immune cells, T cells, that are modified in the lab to better recognize and attack their cancer cells. CRISPR makes that modification process much more efficient and precise. And there's even research using CRISPR to edit the CCR5 receptor in human cells. That's the doorway HIV uses to get in. The hope is to create resistance to HIV infection.
Wow, so direct treatments for genetic diseases, better cancer therapies. What about outside of human health?
Big impact in agriculture, too. Think about feeding a growing planet and dealing with climate change. Scientists are using CRISPR to create crops that are resistant to diseases or pests, which could mean less reliance on pesticides. They're also working on improving crop yield, making plants more tolerant to drought or heat, or even boosting their nutritional value, things like higher vitamin content.
And for scientists just trying to understand basic biology.
Oh, it's indispensable in the research lab now. Need to figure out what a specific gene does, use CRISPR to knock it out, turn it off in cells or a model organism like a mouse or zebrafish and see what happens. Or you can knock in a gene, perhaps inserting a fluorescent tag to track where a protein goes in a cell, or introducing a human disease gene into an animal model to study it better. It just speeds up fundamental discovery so much.
And the future potential,
Yeah.
where might this all lead?
Well, the big dream is potentially eradicating certain devastating inherited diseases entirely. And beyond that, it's a cornerstone for synthetic biology, designing new biological systems or functions. Think biofuels, new materials, bespoke microbes for industry. The possibilities are vast.
Staggering potential, really, from fixing genetic diseases to redesigning crops. But a tool this powerful, it must have downsides, right? Limitations, ethical worries. What's the flip side?
Absolutely. With great power comes, well, significant challenges and very serious questions. A major technical concern is off-target effects. Despite CRISPR's precision, there's still a small risk that the Cas9 might cut DNA somewhere else in the genome, not just at the intended target. These unintended cuts could potentially disrupt other important genes, leading to unforeseen problems like maybe even cancer down the line. Ensuring specificity is still a huge focus of research, making it even more precise.
Okay, so unintended cuts are a technical risk. What about the bigger picture, the ethical side?
Yeah, and that's where the debates get really intense. The ability to edit human DNA brings up huge ethical issues. You hear the term designer babies thrown around, often in a perhaps overly sensational way, but the core question is real. Where do we draw the line? Should we only use CRISPR to treat or prevent serious diseases? What about enhancing traits, making someone taller, smarter, more athletic? Who decides what's a disease versus an enhancement? And could this lead to new forms of inequality?
And that brings us to the really big one, doesn't it? Changing genes that get passed down.
Exactly. The debate around germline editing. This means making changes to sperm, eggs, or embryos, changes that would be inherited by all future generations. Most current gene therapy research focuses on somatic cells, non-reproductive cells, so the changes affect only the treated individual. But germline editing, that permanently alters the human gene pool. The implications are profound. Are we wise enough to make such irreversible changes? Should we? It fundamentally challenges how we see ourselves and our future as a species.
Wow. Okay, definitely a lot to think about there. So just to quickly wrap up our deep dive. CRISPR-Cas9, it began as this really clever defense mechanism in bacteria. And now it's been engineered into this incredibly powerful tool for editing genes in essentially any organism. We talked about the key parts, Cas9 the scissors, the gRNA guiding system, that crucial PAM checkpoint, and how it uses the cell's own repair crews, NHG and HTR. Its speed, affordability, and precision have totally revolutionized research and opened up amazing applications, fixing genetic diseases, fighting cancer, improving our food supply. But as we just discussed, it comes with significant challenges, the risk of off-target cuts and these deep, complex ethical questions, especially around germline editing and what it means to reshape our genetic future.
It leaves us with a pretty profound thought, doesn't it, given this incredible power, this precision tool, CRISPR. How do we, as a society, manage the immense responsibility that comes with it? How do you make sure its benefits are used wisely, shared fairly, while we really grapple with these huge ethical questions about altering life itself, about our future? That's the ongoing conversation we all need to be part of.
A powerful question indeed, and a crucial conversation for all of us. Thank you for joining us on this deep dive into the world of CRISPR-Cas9. It's a field moving at lightning speed, so we definitely encourage you to keep learning and stay engaged with the discussions. Until next time.